Okay, hello everyone. Let's start our mechanics session of the day. The first speaker is Draga Pihler-Puzović, and she's going to talk to us about flows in elastic membranes, elastic Hele-Shaw cells, and how you can assemble a fluidic fuse. So that sounds very interesting. Thank you so much, Draga, for coming. It is a great pleasure to have you here. You can please start sharing your screen and start your talk. Thank you so much.
Thank you, Yuri. I'm just going to start sharing my talk now. Let's see if it works. So can you see it? Good?
Yes, it's perfect. Yes, thank you.
Okay, perfect. So thank you very much, and thanks for making me the part of this meeting. As you remember, I was in Brasilia not so long ago, in 2014, and I had a wonderful time back then, so I was always planning to come back, and I really hope that next time it's going to be in person. So, okay, today I'm going to tell you a little bit about the work that I've been doing in Manchester. And really, this is not just my work, it's a collective effort of a lot of people. I've started working on these problems about 10 years ago when I joined the group of Angel. Back at the time I was a postdoc. We have collaborators in the school, in the Department of Mathematics, and that's Matthias Heil and Andrew Hazel. I was coming from Cambridge, so it turned out that we had loads of common themes to discuss with John Lister and Gunnar Pank, who was in Cambridge at the time. And so they got involved in this project as well. And I'm also going to be showing you results of collaboration with lots of very, very clever PhD students and postdocs. And in particular, that would be Lucy de Clouet, Callum Cuttle, and Finn Box. Okay, so the title for my talk was inspired by Hele-Shaw cells. And this is sort of a Hele-Shaw cell. It's a very simple experimental apparatus. Oh, I have a very sensitive mouse, so apologies for that. It's a very simple experimental apparatus in which you have two parallel rigid walls, which are very close to each other. And then the gap between those walls is vanishingly small, so it's asymptotically small. Consequence of that is that if you consider any flow inside that system, you have effectively a parabolic profile in the direction, in this narrow direction, which means that you can write down the average velocity in that individual cross-section as some constant times the gradient of the pressure field, which is effectively two-dimensional as well, and the pressure field satisfies the Laplace equations. The consequence of that is that flows in Hele-Shaw cells are analogous to inviscid potential flows and to the flows of fluid through a porous media, in other words, a Darcy flow in 2D. And so these cells have been incredibly popular to use in science of geophysics, biophysics, all sorts of other sort of application of fluid dynamics. And in particular, more recently, they have been extensively used in microfluidics as well because of the procedures that are involved in kind of making these microfluidic channels. These microfluidic channels are usually quite narrow, just like these Hele-Shaw cells. So there's a small gap between two walls, and they're usually quite long as well. So there is this separation of length scales in these channels as well. In particular, one of the problems for which the Hele-Shaw cells have been used a lot is to study a two-phase displacement flow in one. So for example, if you're filling the cell with some viscous fluid and then trying to push out that cell, push the fluid out of that cell by injecting a different fluid, which is of a lower viscosity, then at the interface between these two fluids, oh, video stopped playing. Let's try again. At the interface between these two fluids, you have development of the instability called viscous fingering, and this viscous fingering is very easy to actually observe. So here, this is a top view of a radial Hele-Shaw cell where we're injecting air in the middle of the cell to push out some silicon oil. And what you're seeing is that the interface very quickly develops a very complex, interesting pattern. Then you can study in loads of details. What's particularly interesting about this problem is that all of the information about this system is effectively encapsulated by the interface, because all of the nonlinearity which causes the instability, or is there, instability is there because of this nonlinearity, all of it is contained at the interface itself. Elsewhere, the flow is governed by the linear equations that are spelled out there. And so just by looking at this interface, you can study dynamics of this system. What's also interesting about this problem is that it's very prone to instabilities, as you can see one, but it's also, there are lots of signs of disordered dynamics in the system. So if we were to repeat this experiment again by injecting air again into the same system at the same control parameters, you would probably see a different sequence of sort of tip splitting events, finger competition, and so on. And so all of that are signs of disordered dynamics. And finally, this is sort of one of those canonical systems in which people study a lot. And the reason for that is because it is a canonical system for studying morphological growth more generally. So when I talk to my little daughter, who is 5 years old, and she has like her own private Hele-Shaw cell at home, that's how simple the system is. I let her play with these sort of systems. We fill in the gap with some glycerol or some honey lying around, and just she then injects a bit of air into it, and then she looks at these patterns and tells me, "Mommy, we're making a snowflake, a liquid snowflake." And the reason why she's not wrong is because solidification processes actually are described by very similar equations to what describes this morphological growth. So by learning about this system, people more generally learn about morphological growth more generally. Right? Okay, so that's kind of why this particular instability is interesting. But I just want to kind of take a step back and answer the question from point of view of mechanics: why does this instability happen in the first place? So here is the simple mechanism. If we have two parallel rigid walls with the gap between those walls given as H0, then if you're injecting air, you have some constant pressure there. There is some pressure in the viscous fluid. And then if there is no perturbation in the system across that interface between air and fluid, there is a constant pressure drop because there is the interface, right? And the speed at which the interface propagates is effectively given by the same formula that I already showed you before. Now, imagine you have some perturbation in the system. Those can be anything, right? Some imperfection in the fluid, for example. Then ahead of that perturbation, you have a larger pressure gradient, simply because you're closer to the edge of the cell. And therefore, you automatically have a larger velocity at the tip of that finger. As a consequence, fingering grows. So that's why this fingering is called viscous fingering, because the viscous mechanism actually promotes the instability growth. On the other hand, you've perturbed your interface, which means that there is surface tension kicking in to start to prevent the instability from forming, because you've obviously introduced the curvature. If you look at the patterns now from the top, you've introduced some curvature into your system. And so according to the Young's-Laplace relationship, you have a restoring force because of that in your system as well. Of course, the bigger contribution to the curvature would be the one from interface being deformed in this cross-section as well. But unfortunately, that doesn't help you much for this particular system with parallel walls, because that curvature is there whether or not you have a perturbation at your interface. So in other words, you have surface tension force acting, but mainly due to this curvature to help you restore the perturbation. So there is this competition between capillary forces and viscous forces, and in practice, viscous forces easily take over, and so you have a growth in instability. That sort of competition is basically encapsulated by the capillary number. For sufficiently small capillary numbers, you're stable. As capillary number increases in your system, the interface becomes unstable. Now, in this talk, I promised to talk about elastic Hele-Shaw cells. And so that's what I'm going to do. I'm going to, instead of considering a classical system where you have two parallel walls very close to each other, I'm going to put a latex membrane on the top of my Hele-Shaw cell instead of one of the rigid walls. And then I'm going to repeat exactly the same experiment. Try to basically displace as much as possible the viscous fluid that occupies that small gap between a rigid and elastic sheet on the top. Let's see what happens in that case. So here is the experiment that you've seen before with some fingering. And this is the same experiment but below elastic sheet. As you can see in a minute, basically the whole instability has disappeared. And now we have a circular circumference that of the interface that sort of expands as we inject more and more liquid. As you can imagine, while injecting air, we're not just displacing the viscous liquid, we're also inflating this sheet because it's elastic and it responds to the pressure that we've generated by injecting some air into the system. And that's why this particular movie was sped up 2 times. So just so we kind of have time to actually watch it and not spend too much time on this, right? So it's a much slower process. Okay, that's not the whole story. The instability hasn't completely disappeared. It's still there, but you need to inject at a much higher injection rate. And then you will see the instability, and this is how it looks like. It actually represents lots of little stubby fingers that are around, not much closer to where the sort of interface sort of finishes or sits, right? And these short and stubby fingers look nothing like those fingers that extended all the way to the injection point, right? So there is clearly some saturation length in that fingering that happens around the interface. Okay, so why did we start doing this problem in the first place? We embarked on this journey because we were actually interested in a question of area reopening, and we wanted to know. So you might know that newborns are before they take their first breath, born with their lungs collapsed, and so that the lung is collapsed and filled with the fluid, viscous fluid. And then if you look at the cross section of the corresponding tube, it looks a little bit like this. It's basically that's a cross section of the tube filled with fluid, and there is a region where there is a near opposite wall contact. So it's a very sort of narrow region that contains liquid in it. And as with the first breath, this tube gets reopened, the air penetrates into this system and kind of pushes the liquid that's inside and peels apart these walls. So we really wanted to look into that process and understand why does the air go sometimes into this narrowest part of the tube, of the collapsed tube. But there are lots of other systems in which you have similar kinds of flows. Our collaborators in Cambridge, so Gunnar and John, were interested in laccoliths formations. So laccoliths are these hills which are formed when you have magma that penetrates different earth crust, basically layers of earth crust, and it gets to the very top, and then the very top, it doesn't go all the way out, it kind of lifts the very top of the crust, and you end up after some erosion, etc., you end up with these sort of hills. So they wanted to know more about that process, and that's how they've ended up with a similar looking elastic Hele-Shaw cell. And there are other questions, for example, how blisters occur. Similar problems have been considered in literature. So people looked into sort of oil recovery problems, how does oil get recovered in fluid structure interaction systems? How is a roll coating happening? So if you have rolls that typically, sometimes they're coated with elastic structures so that you reduce instabilities, interfacial instabilities that happen there. But also kind of story that I'm telling you today is more generally relevant for all of the studies that explore control and suppression of interfacial instabilities. And there's lots of different ways in which they can be controlled and suppressed. Okay, so that's kind of why we've done it. So now how we've done it. So obviously we've done lots of experiments, but we also done a lot of mathematical modeling. And here is a complete model that can actually reproduce what we see in the experiment. So there is a lot of equations here, but I promise I won't be showing too many equations too often. So stay with me. We're going to stay on this slide for a little bit. So first of all, we have to model fluid. And for fluid, we take this, just like in a classical system where you have two rigid walls, we take the lubrication approximation, and we write down equations for the fluid's depth average velocity and the pressure. And that equation is exactly the same as what I've already shown you in the first slide of this talk. Now, that equation for the pressure, it's different. That equation is different. The reason why it's different is because now the depth of our liquid layer can in principle vary as the sheet responds to the flow. So it's a fluid structure interaction problem, and the liquid changes, the pressure liquid changes as the configuration of the setup changes as well. And that's sort of a first source of nonlinearity, if you want, in our system. We also have to have some boundary conditions at the interface. So this one is just the kinematic boundary conditions saying at which speed the interface expands. That one is just a Young's Laplace equation that I already kind of briefly showed to you, but here I actually spelled out the contributions from different curvatures in the system. So that would be a contribution from the curvature if you look at the system from the top, and that would be a contribution if you're looking at the radial cross-section. Now, this contribution is a lot more important in this system compared to a parallel walled Hele-Shaw cell, because of course now, as I was saying earlier, because the sheet was deforming, you can potentially have different contribution to the surface tension and different parts of your cell. All of this then needs to be coupled to what the elastic wall is doing, and that's what's happening here. And what we observed in our experiments is that our elastic walls were not just bending, they were also stretching. And so we kind of couple these equations to the simplest equations for the thin membranes that did the both stretching and the bending behavior. Those were Foppelman-Common equations. So they are written here, and they're also nonlinear equations. So the terms that's just a term that corresponds to the vertical displacement of the sheet. We have also this phi, and phi is an area stress function, which allows us to compute the stresses in the cell and in the elastic wall. And all of that is obviously coupled to what the fluid does. So that pressure is the same pressure as in fluid. These equations, the nonlinearities, these equations are contained within these curly brackets, which I haven't spelled out just to save some space, but you should be aware that these are nonlinear terms. And we also have some boundary conditions as well that we need to solve. All of that needs to be closed. This problem needs to be closed by including some mass conservation as well, equations into it. And finally, the one thing that I want to point out is because we had experiments, we were really after the quantitative agreement between experiments and theory. So we knew from the start that this model would not yield it unless we can somehow count for very thin films that were left on the top and the bottom wall of our Hele-Shaw cell as the bubble was expanding. Of course, this approximation does not do that because the equations are dealing with the fluid depth average, velocity, and pressure. So if you just stick to this lubrication approximation, this is what you're effectively modeling. You're not modeling the fact that there is some layer of liquid left at the back here behind the bubble tip and at the top there as well. So in order to account for that, we've introduced some ad hoc changes to our model, and we've basically corrected our interfacial boundary conditions to account for these films. And we've done that. There is no rational way of explaining this other than we've done it by fitting to the numerics that were published back in the 80s. But we did verify this against the Stokes equations in particular regimes. And we also, we have very good quantity agreement with experiments. So we were happy with the ad hoc changes that we made to our model. And so what we were modeling with this is an expansion of the bubble in elastic Hele-Shaw cell, which can account for these thin films left behind as well. Okay, so that's the set of equations, the complete set of equations we have for this problem. And then we started analyzing it. So we've solved for this set of equations assuming axisymmetric expansion. We also did linear stability analysis by perturbing those axisymmetric solutions non-axisymmetrically, and then computing the growth rate of the instability. So by looking at this growth rate, we can basically conclude that instability decays to zero. The perturbations decay to zero if the growth rate is negative. If it increases in time, then the instability grows. And so that allows us to actually characterize the onset, at least the onset of the instability. And then we also compare all of that to direct numerical simulations as well that we've done using UNFLIP, which is our in-house built finite element library that deals particularly well with these kind of problems where you have a moving interface, such as the one in here. And here are some examples of the bloom flip simulations. So in direction of an arrow, we increase the wall elasticity, starting with the rigid wall. So this is a classical rigid Hele-Shaw cell, and then ending up with a very flexible wall. And as you can see, at least qualitatively, we reproduce what we see in the experiment. So we go from complex fingering to some much shorter stubby fingers near the interface, and finally to suppression of fingering altogether. Okay, so we've done all of this analysis, and we actually worked on this problem for about 10 years now, as I have said. So there's a lot of maths and a lot of numerics that's been put into this problem. So for today, I thought I'd just kind of summarize the main ideas in physics that we've learned based on that math, and hopefully use some relatively simple mathematics arguments to show you why we made those conclusions. So let me just remind you that what the mechanism for instability onset was in a rigid cell. So it was this competition between surface tension and viscous forces, and effectively viscous force, when the viscous forces would dominate, you would see instability development. Now in the elastic cell, you have a potential, so things are different. You now have another free surface, the fact that the elastic sheet can also deform. And so you might expect some contribution from that. But this is what turns out to be the case in our experiments. As we inject air at the constant rate, we inflate our sheet as well as displace our fluid. So that automatically means that our local capillary numbers, so the speed at which the interface expands, it's already lower than compared to the analogous rigid cell. Right? Okay, so that's the first conclusion that we can make. We already have that on our side. We expect just from there the interface to be more stable. But another interesting fact is that as we are inflating our sheet, we are also so our interface also suddenly starts penetrating into something that looks like a tapered cell. So we're no longer having a situation where the interface is flowing in a cell with a constant depth, but actually goes into some sort of a wedge of the fluid that's ahead of that interface. And that's very important because it turns out that everything that's happening in this problem is due to the fact that we have that wedge. Let me elaborate a little bit more on this. So here, I'm just showing you the results of, to begin with, the results of the axisymmetric simulation for one set of parameters in our cell, and for 3 different instances in time. And so what you're looking at is the sheet deformation, as well as the position, the corresponding position of the interface. And what you can see here is that, first of all, this sheet deformation, the sheet is kind of quite a bit inflated behind. There's a little bit of inflation in the front, but effectively all of that deformation is more or less constrained to this wedge region that I've drawn there. So the deformation of the sheet quickly decays to 0 ahead of the interface. Interestingly, if you look at the pressure as well and so on, you will notice that actually all of the action again is happening in this wedge. So very quickly the pressure decays to 0 far ahead of the edge, and effectively what that means is that you have a redistribution flow in that region, and not much flow beyond that region. Okay, so that's the first observation. The second observation, what could do is by looking at, so this was of course all for just axisymmetric flow. So now we can look at the perturbations to that axisymmetric flow and look at what the perturbation terms do. And this is just plotting those perturbation terms, for example, for the sheet and the pressure. And again, we've just scaled them here. So they're just scaled here just so that they can be shown on the same, more or less same plots. But what you're looking at here is basically that all of the action, once again, is constrained to the wedge region. So the perturbation to the sheet is pretty much there. And in practice, turns out to be negligible. In fact, the numbers are so tiny. And the perturbation to the pressure field, again, is again in this wet region. And it quickly basically decays to 0 beyond that. Okay, so in other words, everything is in there. One last thing I want to point out, so all of this is consequence, all of the results that I'm showing you here are linear stability analysis results. I just want to point out that if you, on top of that, plot some experiments as well, and here I'm just showing where the fingers, those stubby fingers sit in the experiment. The left-hand side of these lines here is where they start, the right-hand side is where they finish. You can see that all of the fingers, once again, sit in the wedge region predicted by the axisymmetric numerics. So again, another evidence that everything is happening there. Okay, so in that case, you might ask, okay, so what is then the mechanism for the suppression of instability or delay the onset of instability? Well, because we have this edge, this wedge ahead of the interface, what we are doing here effectively is propagating into something that looks similar to the rigid but inclined wall. And that means that we no longer have a constant layer of depth h0 ahead of the interface, but it's actually some layer of depth h that might vary and in fact decreases as we propagate further and further into wedge. So that means that we have two additional mechanisms to help us stabilize the interface. The first one is due to the transverse curvature that we could completely neglect before in terms of stabilization. So let's see how it works. Imagine you have an interface and you perturbed it, like we did here. That means that perturbation moves into the region where the depth of the cell is actually smaller than the depth of the cell behind. Right? So that means that the curvature is bigger there. And that means that the restoring forces due to surface tension are actually more significant for that part of the interface than they are for this part of the interface. And so you have additional stabilization going on there. That's one mechanism. Another one is due to viscous forces. Effectively, you're pushing, again, this perturbation is in a region where h is smaller, and therefore, according to just this relationship, your speed there is automatically smaller than it is behind. So your viscous forces turn out to be less destabilizing in this geometrical configuration. So combine these two effects, plus the fact that you are overall slowing down the interface because you're inflating, all combined, they contribute to the suppression of instability. And that's why we see suppression instability, such a dramatic suppression instability in our experiments. Okay, so we want to say a few more words about the stability itself beyond the onset. And just to kind of show what we can say, I thought I demonstrate it using experiments. And here is an instantaneous picture of pattern. And then we can characterize that pattern both from our numerical simulations as well as from our experiments by looking, say, at the depth of individual fingers that are formed. And if we, say, plot the depth of that finger as a function of the average radius, which is in this case the proxy for time, what we'll see is that the depth goes up, reaches a certain value, and then starts going down again. So what this means is that our instability under elastic sheets is actually transient. We never see a pattern that is basically saturated and just sitting there. It's by its very nature a transient instability. And just by looking at the size of these error bars, you can see there is quite a bit of noise in the system as well. I mean, if you take a very careful look at different figures, you will see that they're all at slightly different stages of development, which means that, combine these two pieces of information are a sign of a bit of a trouble, because you can never be 100% sure that you're characterizing something that is a long-term state, right? Everything is transient, and so whatever you say about any part of this evolution, you can argue, well, but you're saying something about that's a transient and therefore potentially not a real effect, right? Okay, so because of that, we're adamant to say something, that we focus on the particular sort of stage of development of this fingering. We focused at the point where the fingers would reach the maximum depth before they would decay. And then at that point, we started looking at different patterns. And here, there are lots of different experiments that we've performed. by varying sheet stiffnesses, by varying the thicknesses of the sheets, and therefore the Young's modulus, the bending modulus of the sheets, and by varying initial depths of our cells. And for all this data that we have, we did start to notice some systematic trends in how the patterns look like. So one thing that I want you to notice here is that the number of fingers, if we say compare the extreme patterns, that one and that one, the number of fingers is clearly different for different patterns. So here you have a lot more fingers and they're a lot less deep, right? They're a lot shallower than they are in here. Here you have a few fingers, but they're deeper, they're further extended towards the injection point. And so when we summarize all of this data, just at the point before the instability was decaying, what we observed is these green points here, basically. And then we've predicted some wave numbers at that stage, and then we compared all of that to our linear stability analysis as well. So we said, okay, let's now compare. We appreciate that these are nonlinear patterns just before the stabilities are decaying, but let's now compare it to what our linear stability says should be the most unstable wave number at that stage. And surprisingly, even to us, our linear stability analysis predicted exactly the same wave numbers. So those are shown with these red lines. As you can see, it worked so well over the whole range of parameters that we were, by the end of the exercise, convinced that the wave number is set by the linear stability analysis, even though in principle the instability is transient, and given enough space in the experiment, it would eventually decay. While it's there, it's all to do with the linear stability analysis. Okay, so that was the first observation that we made. The second one that we noticed was this idea that all our fingers also kind of lined up in terms of how deep they are with respect to the injection point. So here, they're really, really shallow. Again, I want you to look at the extreme cases. So they're very shallow here, and they're much deeper for this sheet, for example. And so what we've done is we've, again, looked at the this depth of the fingers as a function of all our parameters, and here it is shown with points. And then on top of that, we also plotted with the gray the size of our wedges. So remember the wedge being that region where all the recirculation flow was happening. So as a result of that, we found a correlation between the finger length and the length of the wedge. And basically what ended up happening according to this kind of analysis is that we thought that the nonlinear finger saturation was acting on the same length scale as the wedge. But of course, again, you can always dispute these results because everything is transient. And so ultimately, to confirm that the nonlinear patterns were indeed set by this physics, we needed to get into somehow to flows where everything was basically moving steadily, right? And then we wouldn't have transient behavior, so we'd be potentially able to get to saturated nonlinear states. So achieving such flows in a radial geometry is very hard. It's impossible at a constant injection rate. And the reason why it's impossible in a constant, when you're injecting at a constant flux, is from simple sort of geometry. You can imagine as your interface extends, your circumference becomes bigger and bigger and bigger. And because the flux is constant, you therefore have to kind of slow down to accommodate bigger and bigger area that you're occupying. So we knew that this would not work in a radial cell, but we still wanted to. We were still adamant that we could make it work, and the reason why we thought that was possible because we actually got inspiration from these two sort of classical works. So the first one was by McEwen and Taylor, and in which they were peeling the sheet of a solid substrate, which was separated from the sheet with a bit of a viscous fluid. And so they would literally just taking the sheet up at some constant rate. And there you effectively have a speed, you have an interface that propagates at the constant speed. And back in 1966, they also reported an instability that looked very similar to our fingers, But of course, but at the time they didn't characterize it in any depth. They just showed those pictures. But of course, that instability is in their scenario not transient. It's there and it then moves at a constant speed as long as they do the experiment. The other sort of version of this is classical printer's instability. So if you have two co-rotating cylinders with the gap between those cylinders filled with some viscous fluid, then if they're co-rotating at a certain speed there, by design, there is some air that's protruding that space between the two cylinders, and therefore there is an interface between air and the viscous liquid, and that interface goes unstable due to the same sort of primary mechanism that I've explained already. And in that version of events, when the interface goes unstable, the instability looks very similar once again to our fingering instability in elastic Hele-Shaw cells. So there were examples out there of saturated patterns that move at constant speed, and we thought, clearly we should be able to kind of demonstrate there is a similarity, more decisively there is a similarity between our patterns and these patterns. Radial cells were not an option, so we went to rectangular cells, rectangular Hele-Shaw cells. Now, in rectangular Hele-Shaw cells, when you're injecting at the constant injection rate, when you have two parallel rigid walls, you also cannot sustain a flat interface. The reason why you can't sustain a flat interface is because you have the fingering instability. And the result of that fingering instability, you have a single finger that develops and moves inside this channel and has a particular width and a particular curvature. That particular width and the particular curvature are set by channel aspect ratio and the capillary number. And so, if you have that configuration, there is no hope to study again our viscous fingering, stubby viscous fingering, but at least you have a steady propagation of the front. Now, we then went and designed an elastic-walled Hele-Shaw cell, Hele-Shaw channel, in which we basically took this classical Hele-Shaw channel, but replaced the top wall with the elastic sheet, and that was pre-tensioned and clamped in place. And then we tried running the sort of displacing the liquid inside that cell with some air, and indeed we were able to obtain steadily propagating interfaces. So I'm not showing you the movie, but I'm showing instead the pressure trace in such experiment. And you can see that after some transients, our pressure in the bubble was more or less constant, and then you would see some end effects. So in other words, in this region, we did indeed get a steadily propagating interface that we could then explore. Another thing to remember, that of course, as soon as you have an elastic sheet on the top, you have another degree of freedom in your system. So you could potentially try to play with the cross-section of your system before you start injecting any fluid or any air. And you can do so by adjusting the pressure difference between the inside the liquid and the outside, and generate all sorts of initial levels of collapse as shown here. So the reason why we did all of this is because what we were after was a bubble in which we can somehow generate a flat interface that moves at a constant speed. So in order to do that, and this really comes from lots of experience that my collaborator, who was the PI in this project, and Jules has with working with these sort of bubbles. So in order to achieve the bubble that moves at the constant speed and is flat at the front, what you need to do is move to in parameter space to a region of high Reynolds number and high initial level of collapse. And in that case, you can get a flat tip finger. And if you look at the center of that channel, then as the finger propagates into your system, you generate the same kind of geometry. So you generate the same kind of taper or the same kind of wedge as in elastic Hele-Shaw cells that I've shown you earlier with the radial geometry. But what the great benefit of this is that now the front is moving at the constant speed. So therefore, if you can generate some instability on top of this front, you can also get that instability to saturate and not be transient, and therefore explore sort of nonlinear patterns that are formed in this system. Okay, so that's what we've done. One more thing I should mention and give all the credit to Anne for is the fact that we actually did a little bit more than just use these channels with the initial high level of initial collapse. We've actually replaced them for better control of initial thickness in our system, of thickness of the viscous layer of fluid in our system. We've actually, instead of collapsing the channel, we replaced that by putting in occlusion into our channel. And this was done so that we can sort of mimic the high level of collapse, but also generate these flat tip bubbles, which then we can then study the secondary fingering instability, stubby fingerings on top of. And if you do this in engineering magic, basically, you indeed end up with these stubby fingers on top of flat fingers. And this is what's shown here. And then you can vary all your parameters in the system almost independently by being sort of a genius engineer, basically. And so in particular, you can kind of generate the system in which the angle of the wedge can be kept constant, but then you can kind of change the flux, for example, thereby changing the capillary number at which the finger propagates. And as a result of that, get sort of fingers of different depth to study. And so here is, so all these experiments were done by Lucie Ducloën, who worked with us at the time. And she plotted the results of those experiments where she looked at the depth of these fingers onto a same curve. Basically, all these experiments collapsed into the same curve where she was plotting the depth of these fingers, the length of these fingers as a function of 1 over the tangent of the taper angle. And all the data collapsed, basically suggesting that the finger length was scaling like 1 over the tangent. So what does that mean? That means for a given initial depth of the liquid, our patterns extend into critical. So just if you do the simple geometry, basic trigonometry, our patterns extended to a critical height of reopening and did not go any further. And so again, this is another confirmation that all that's happening is you are redistributing, when forming these patterns, you're redistributing the liquid that's contained within this wedge, and that's effectively controlling this process. But now we had sort of a hard evidence that that's indeed the case, because these behaviors are were no longer transient in our experiments, and we could sustain these patterns throughout the range of the throughout the range of the channel where this behavior was observed. Okay, so just a final note on this, I want to say is that, I said this really provides us with the means to link two classical interfacial instabilities. And when you think about it, viscous fingering is just like the fingering that I've described here, except between two parallel walls. So that means that there is no taper and your angle is effectively 0, which means that according to this relationship, your length of your fingers should be infinity. And indeed, they do sort of extend all the way almost to the ejection point in these images. On the other hand, if you have a printer's instability, you are by design redistributing the final amounts of fluids. And so you fall, depending on how much fluid you have and what the angle of reopening is, you fall onto one of these, basically onto different parts of the same curve. So in other words, you're dealing with two sides of the same coin, and then really there shouldn't be any distinction between viscous fingering and printer instability. They are the same instability, just in different regimes, if you want. Okay, so just a final note, we've studied, we got interested in these elastic walled Hele-Shaw channels as well, and we studied lots of pattern formation in those channels. I'm not going to talk about it. I just want to show you images of how beautiful these things can get. And here, this is just a spatial temporal image of what's going on there. And because the maths and is so elegant and it's all relatively simple, you can actually relatively simply predict even these kind of streaks that form of liquid between different parts of the finger. But of course, if you want to be more precise than that, then one needs to do some simulations of the system to predict quantitative behavior. And that becomes a very, very complicated problem that has been actually tackled by one of the PhD students in our group, who happens to be Brazilian and is currently still based in Manchester. Okay, so I'm going to move on to a next part of my talk in my last 10 minutes. And what I want to talk now about very briefly is our elastic Hele-Shaw cells with soft substrates in them. So now, instead of having a thin elastic sheet on top of a rigid wall, what we've done is we've replaced the bottom rigid wall with a mold that contained a very soft material in it. So think jelly that you use to make food and cakes and stuff. So that's how soft it was. The reason why we started studying this problem was because we were really intrigued about how different the deformations would be in this system. And when you think about it, there is a fundamental difference between this system with the elastic wall and the system that I just talked about before. The main difference is that in the system before, we have never constrained the amounts of liquid you can accommodate or fluid that you can accommodate within the cell. So by injecting air, you were able to kind of displace the elastic wall as much as you would like to, and the overall volume of the Hele-Shaw cell was not the same at the start of the experiment and at the end of the experiment. Here, that's not the case because we're using incompressible liquid. We're using an incompressible material for this soft wall. Effectively, the total volume of the cell is at all times conserved. So we were expecting to see a very different behavior in this sort of system. And to begin with, we thought we would kind of start by just classifying the corresponding deformations of the thin wall. And so we've simplified our model. We removed the interface from the system and just started injecting the same viscous fluid that occupies the cell to begin with, and looking at what are the corresponding deformations of the wall. So here are our observations. So let's start by monitoring the substrate deformation. So we're looking here at the substrate deformation versus the position or how far you are in a cross-section of the Hele-Shaw cell. And so substrate deformations are shown with these lines, and all the vertical lines are positions of where the intruding fluid is at all times in the experiment. And if you start with a relatively stiff substrate, then not much has happened really, or not much happens. You have a relatively rapid sort of deformation in the center of the cell where you're injecting the liquid, and then very quickly it's sort of saturates to some shape, steady shape, and then as the more liquid arrives, that it just spreads out, and that's pretty much it. Now, if you take much softer substrates, then things get a bit more exciting because you are not just deforming your cell in the middle, but there is some deformation going on near the rim of the cell as well. And so just remember that there is this bulging going on, but in principle, you also kind of very relatively quickly attain these kind of shapes where you have a bit of bulging at the edge. Now, these are the first experiments that we've done. And then we started monitoring, the postdoc who was working on this, Finn, started monitoring more widely what happens is you vary lots of parameters in the system, for example, the depth of the cell, the thickness of the substrate that you use, and so on. And he was, just to save time, monitoring the the position of the intruding fluid with respect to injection point. And so at 1, you basically reach the edge of the cell. At 0, you just start expanding. And that is the function of time. And what he saw is that, as he was injecting more and more liquid, the liquid was at different rates. Liquid would eventually escape the cell in most cases, apart from a specific set of cases which are shown in black here. And those were actually the most interesting cases that I want to discuss now. So if you just follow those black points, you see that initially, so the injected liquid into the cell just expands happily with time, but then eventually kind of reaches the point at which there is no longer any expansion, and basically the curve just flattens out. And at those points here where the curve flattens out, our whole experiment breaks down, because our syringe pump that we use to inject our liquids with stalls at this stage, right? And it doesn't work. The system basically breaks down. So let me show you in another sort of representation of that in the following movie. So this is now looking at this Hele-Shaw cell from the top, and just so that you can visualize the injected fluid, which is of the same kind. So there isn't a real interface here. It's just the fluid that's getting injected is colored. You'll be able to see where it goes, basically. Here it is. So this is a colored injected fluid, but it's of the same kind as the one that is getting displaced. And what you can see here is that basically, for a while, this was expanding, this front was expanding, and then eventually got stuck. It was just sitting there, and it couldn't expand anymore. Okay, so what happened here is that the flow somehow choked itself. Right? It could not be sustained, and our syringe pumps stalled, and the whole experiment was interrupted. And we really wanted to understand what was going on, why was this going on. So we went to model this. We went to model this, and we modeled this using a mathematical model, which is very similar to the mathematical model that I explained before. So it's the same kind of lubrication approximation for the fluids flow, and that's combined with the boundary conditions that come from the mass conservation. Of course, the only difference in the fluids now is that this set of equations is actually simpler because we don't have any interface. It's the same fluid that's flowing through the cell all the time. And then that set of equations has to be coupled to the equation of the elastic wall, and now we have a fixed slab of material. But to begin with, we just assumed linear elasticity and brought down the equilibrium equations for the stress and displacement, which were, in this particular case, a bit more complicated, and they're two-dimensional as opposed to one-dimensional in the membranes. And then we applied some boundary conditions. So we assumed that basically there was a no-slip around the edges of the cell, and we assumed that where the fluid meets the elastic wall, we have the stresses which are equal to each other, and we have displacements which are equal to each other. So now if you non-dimensionalize all of this, you end up with two non-dimensional parameters, but only one. So the first one is geometrical, and I'm not dwelling on it too much here. I want to stress the second one, which is the fluid structure interaction parameter, which relates stresses to elastic stresses in this system. And this turns out to be the key parameter for explaining why we have this choking flow. The clue for why the choking flow would occur is really in this picture that I already showed you. And it's all to do with this bulging that happens very near the rim of our cell, where the cell essentially ends. And this is the story. In this region where the bulging happens, we effectively have an elastic boundary layer, which is in the order of the thickness of the substrate. We must satisfy the equations for the flux at that point, which come from the lubrication flow in that region. But also, we can imagine that as our membrane is deforming, as our substrate is deforming, at some point it's going to deform so much that it's going to touch the top boundary of our Hele-Shaw cell. So we can estimate the pressure needed for this to occur. We can estimate the pressure needed for our elastic boundary to deform in such way so that it basically touches the top cell, and that's what's happening here. And then by balancing the pressure in the fluid to the pressure that's needed for that to occur, we can extract the value for this fluid structure interaction parameter when we explain what we expect our elastic wall to effectively shut down the flow and kind of close off the gap near the edge of the cell. We've obtained that so according to our simple analysis, that value needs to be on the order 1. And then we decided to plot our experimental data on the graph and show whether or not this indeed works. Is this the physical explanation of what's going on? And indeed, we obtained all our data. So this is all our data for when. So we're looking at the bulge versus this parameter f. And all this experimental data, when we had some flow and we could sort of find the size of the bulge, was sitting on the left-hand side of f equals to 1. And then all of the choking events were effectively on the right-hand side of f equals to 1. So in other words, if this parameter is below some critical value of order 1, we can sustain the flow in the system. And otherwise, the flow gets shut by the fact that there is this bulging happening at the edge of the system that shuts down the flow. And mathematically, what that corresponds to is that you have some steady solutions, sort of stable and steady solutions in terms of the formation in the system up until that point. And then at that point, you no longer have a steady solution anymore, and the bulge just grows indefinitely mathematically, and in principle your model just breaks down, and that's what's happening beyond that critical f. Okay, so that's our mechanism for choking the flow. And in my last minute, I'm just gonna, so we observed it, we didn't expect it to happen when we started this experiment. And so once we found it, we thought, well, this is very neat. It's kind of a flow that stops itself. So what's the use of it? And then we went and thought about what could be potential uses of these things. So at the very beginning of this talk, I started talking about sort of microfluidics and saying that these Hele-Shaw cells can potentially be relevant to microfluidics. And so we've started digging into microfluidics literature, and it turns out that there is a lot of literature on passive control in microfluidics. So if you're not familiar with the field, just briefly, I want to show you what microfluidics is this lab on chip where you basically make little channels in which you can float small amounts of fluids. And typically the way you do this, and these are used for all sorts of analysis of flows, etc. But the way these typically run is through lots of external actuators. So here you can see how many syringes you need to kind of generate the right kind of flow on those scales. Now, more recently, people have started looking into sort of passive ways of controlling flows in such systems. And so here we were kind of in this position to be able to maybe suggest how one can use these bulging type deformations to design something like a fluidic fuse that would shut the flow beyond a critical, say, flux in the system. So just the last slide, I want to show you that indeed it does work. We went and tested our theory. So remember, we have this critical value parameter when we get the choking. So that means that we can, in principle, for the given geometry, have a critical flux at which the system stops. Which is the Hele-Shaw cell can no longer sustain the flow and it shuts itself. And so we've tested this idea using water on a microfluidic scale. The reason why we did it with water is that's the most used liquid in microfluidics, and obviously we wanted to use a microfluidic scale just to demonstrate the principle that it would work there as well, that it's not scale dependent. And here what I'm plotting is the outflow outside the cell versus what we put in. And if there is a flow, one would expect the two to be identical to each other as they are. But then at a particular point, the flow shuts itself, and so the outflow goes to 0. And if you then rescale this data, you can see that that point is exactly corresponding to the sort of critical Q that we predicted using this theory. So in other words, we are quite confident that we have something that could, in principle, work on a microfluidic scale. Okay, so that's the end of my talk. I just want to conclude by saying that I've shown you two very different uses of Hele-Shaw cells. with elastic deformable walls. And in the first one, it was all about fundamental fluid dynamics. In the second one, I tried to kind of show you a neat phenomena that could potentially have applications in microfluidics. There are loads of criticism regarding the second sort of application that you could direct in my way. One could be, well, surely in microfluidics, you're more interested in multi-phase flows as opposed to having just the same fluid being injected constantly into the system, and totally take that criticism. But I just want to leave you with this movie of what happens in the same Hele-Shaw cell if you have an interface in it as well. And if you do that, then you can still get the choking events. And here is an example of a choking event when fingers cannot escape the cell. So the cell has shut itself down, and you can kind of, if I keep playing this movie forever, you will see that it kind of sits there basically because the cell is choked. Okay, so thank you very much for your attention. I'm sorry I'm slightly overrunning. If you're interested in any of the details or any of the maths of what I was talking about today, all of that is published in these papers, so please check them out. Thanks once again.
Excellent. Thank you so much, Draga, for this very nice talk. Loads of interesting phenomena that you have unveiled to us. Very fascinating. So we are now open for questions. If you want to ask a question, please raise your hand. While people decide whether or not to make a question, I have a quick question, Draga, to you. So you said that the linear stability analysis set the wave number of the instabilities. So when you do the linear stability analysis, what are the mechanisms that appear there that define the number of fingers that you're going to get on every system?
Yeah, so it's the same. It's interestingly, it's the same mechanisms as on the onset, right? So those are the same ingredients that would, in some rather complicated way, contribute to this, right? So it would be, so the control parameters, let's put it that way. Control parameters in the system are a capillary number. Mm-hmm. So that would be the first one that would define what's going on and what's the actual wave number. The other one is a fluid structure interaction. So just like in the second half of the talk, there is a similar fluid structure interaction in the first half of the talk. So there is the parameter that controls that. There is a parameter that controls the relative ratio of bending and stretching forces to each other. So that parameter also will affect the number of fingers you see. And then the final one is geometry itself. So the geometry, there is basically the aspect ratio in the cell. So how big is your cell versus how narrow was it to begin with? So those are the four parameters. And now a certain combination of those affects through the mechanisms at the onset, because the same mechanisms that work for the onset would in principle be there in the linear stability analysis as well.
And the same applies to the ones to the channel fingering that you showed.
So at the channel finger, we've only focused on the nonlinear patterns, right? So there we haven't looked at actually what sets the wave number. There we were just focusing on the depth of the fingers themselves. And that's why because nonlinear, but we haven't actually, that's a good question. We haven't looked at what sets the width necessarily, because the linear stability analysis becomes really complicated in that system.
Yeah, I think the base state would have to be the finger, right?
Yeah, the base state is the finger, exactly. But that's something that Joao, who is our PhD student, he didn't work directly with me, but we co-authored one paper, which is kind of along those lines. He's actually spent his whole PhD trying to find these base states and trying to understand when they go unstable, and it's very complicated.
I bet,
very interesting.
Yeah.
So yeah, it's that's the difficult part. Base state is difficult in that case. So I guess radial versus rectangular geometry each have their own benefits.
Yeah. Very good, very good. So does anyone have another question? Please raise your hand now. So I still have another question if people are still deciding to ask questions or not. So it took me a while to realize why when you show the very first movie on the elastic wall, if you impose the same flow rate, why was it so much slower on the elastic case than in the rigid case. And then I realized that, of course, we're inflating the thing, so some of the volume that you're pumping is actually being spent or is stored at the elevation of height of your membrane. So the question that I have is the membrane that you place on your Hele-Shaw, there is a minimum layer of fluid, right, between the wall, the rigid wall at the bottom, and the elastic layer. Is that right? There is a
I
very
mean, to begin with, there is a very narrow, yes. So it starts just like in the classical system. It starts
right,
with a small layer to begin with,
right,
very thin layer between the two, and then as you inflate, you still have some liquid left. Those are the films that I mentioned in the
right,
model. You still have a little bit of film left on top and the bottom. Yeah,
right. So
absolutely.
so when you increase the elasticity of the rigidity of your wall, so
Yes.
what happens? Does it bend like a triangle, or does it all bend and level up as in a rigid wall? So is it so the question I'm wondering, is it a linear or is it a singular transition from the elastic to the fully rigid system that you are observing? Because in the rigid
Yeah,
system,
it's a really good question. I suspect it's singular. I don't know.
hmm,
We've never explored that, but I suspect it's a singular thing. I don't know. It's an excellent question. It's a bit like the question of what happens if you have a surface tension and you don't.
hmm,
And I
hmm.
suspect it's in there it's a singular limit. I
Yes,
know for
yes, yes.
sure. So here it's just like another free surface. So it's a bit like it's not quite the same effect as surface tension, but I would suspect it's a singular thing. But
Right.
it's an excellent question. I have no idea,
Yeah,
Yuri.
because it would change the way that it responds, right? If you
Yeah.
inflate, the thing would be like triangular, popping up because it's so rigid to bend, or would it just raise everything up and form an equivalent rigid
I guess
wall?
it couldn't raise everything up because I think it would still do that thing, but I'm not sure how the precise transition,
Mm,
if you're epsilon away from the rigid versus exactly the rigid, I think that the rigid uh, is a singular limit in a sense. So there is a
mm,
complete disconnect in behavior in
yes,
the rigid system and the, but it's a very good
because the
question.
elastic layer is clamped as well, right? So that changes the thing.
Well, not in the radial cell. So it doesn't have to be because in the
Ah,
radial cell, the whole there isn't much flow ahead of the interface. It's all in the wedge.
I
So you
see,
can
I
just
see.
leave it be, and whatever you want to do. As long as the boundaries are sufficiently far away, you don't even have to clump it. It can just sit there. That's
Right,
it.
very good. So we have a question on the chat. The Rafael Oliveira. Rafael, if you want to say something, you are very welcome to unmute yourself, or I can just read your question. That was a very good presentation. What was your motivation to study this kind of phenomenon? I've been studying about contagious diseases, and I reckon there are similarities with your studies. How can I access the article you wrote? So I think
No,
you
thank you very
placed
much.
your references, right, at the end of the talk.
Yes.
But Draga?
So I think to access them, I can if you email to me with I can send you preprints. Otherwise, it would have to be accessing journal website. But and there is all the information about those publications. It's on my personal website as well. And Yuri is recording this talk, so you'll be able to read
Yes.
out the specific references that I've put there. As for how was it motivated? So for us, we started with biophysics. We started with this question. We were really interested in like phenomena of error reopening from mechanics point of view. But then it ended up being kind of almost these figuring instabilities are really fascinating from the nonlinear dynamics point of view, and as I said at the beginning of this talk, all the nonlinearities are in the rigid system are at the interface. So that's why nonlinear dynamicists have been interested in these kind of problems and have studied a lot, questions like earlier today in the plenary talk, we heard about ideas of chaos and transitions between different unstable states and stuff like that. So in principle, there is actually some indication that these questions can be asked in the system as well. And there is some disordered, there's definitely some disordered dynamics going on. I wouldn't go as far as saying chaos, but there is definitely disordered
Mm.
dynamics. And so, yeah, we kind of maybe got sidetracked a little bit from our initial motivation, but our initial motivation was definitely just to understand. We started simple. We wanted to understand basically the error reopening problem, and that's what the setup was kind of constructed by Andrew, who started on this project. And then I joined Manchester as a postdoc at the time, and we've started doing all these things.
Very good. So I think we'll have to stop here so that we are not too late in our session. Draga, thank you so much for such a wonderful talk. Very motivating, very impressive with all the results, all the analysis, all the experiments. Congratulations to you and to your group. I hope to see you again soon in another talk, and I hope in person at some time.
Thank you very much. Stay
so
safe.
now we go ahead.
I was just saying, stay safe.
Yes,
See
please.
you.